As soon as a messenger RNA molecule is transported from the nucleus to the cytoplasm, ribosomes begin to translate the sequence into amino acids. Typically, many ribosomes translate the mRNA simultaneously. Each ribosome begins at the 5' end of the mRNA and progresses steadily towards the 3' end. New ribosomes attach to the 5' end at the same rate as the previous ones move out of the way. These multiple initiations allow the cell to make much more protein from a single message than if one ribosome had to complete the task before another could begin. When a ribosome reaches a stop codon, the ribosome and the new protein dissociate from each other and from the mRNA. This electron micrograph depicts a membrane-bound polyribosome from a eukaryotic cell.